I'll just... Can I request everybody for a group photograph, please? We want to... नहीं आ रहा नहीं. हाँ अब I'll check.
कोई बात नहीं, यहाँ से बोल रहे हैं मैम, यहाँ से बोल रहे हैं.
We want to capture this moment in history. So can घंटी ले घंटी लेके आ जाओ.
वैसे ही बोल देते हैं मैम. अच्छा नहीं है.
Check.
Can I request you to kindly be seated? We are that hall is not available. Hall. We are running a little late. Can I request you to kindly be seated so that we can start the next talk? May I request Professor S.K. Mittal and Dr. B. Sesikaran to kindly chair this keynote lecture by Professor Colin Hill. Yes. Professor S.K. Mittal is currently the Chairman, Department of Pediatrics, Pushpanjali Crossley Hospital, Ghaziabad. Professor Mittal is a member of several councils, societies and academies. He is a recipient of several awards, including the Delhi State Award. He has more than 150 publications in indexed journals. Dr. B. Sesikaran is currently the Chairman of Food Labelling Committee of the Food Safety and Standards Authority of India, Chairman of Review Committee on Genetic Manipulation in the Department of Biotechnology, Chairman of Food Hygiene Committee in the Bureau of Indian Standards, and former Director, National Institute of Nutrition, Hyderabad. Dr. Sesikaran is a fellow of several national and international academies. He has received several awards and has more than 100 publications in peer-reviewed journals and 5 book chapters. Can I request the chair to kindly introduce Professor Colin Hill?
Welcome back, and we have great pleasure in welcoming Professor Colin Hill. Professor Hill is a Professor of Microbial Food Safety, Department of Microbiology at the University College at Cork, and is President of the International Scientific Association of Probiotics and Prebiotics, and the member of the Royal Irish Academy and the Fellow of the American Academy of Microbiology. He received the prestigious Elie Mechnikov Prize in 2010. He has over 400 publications and 380 peer-reviewed journal papers and contributed to 30 book chapters. Now I invite Professor Hill to deliver this talk.
Good afternoon, and thanks to the organisers for the invitation and for the support to come here to this meeting. I'm very pleased to be here in India. It's just my second time. And I was lucky to come at all because we have had a hurricane in Ireland, which is very rare, and it stopped just for a few hours and I managed to get a flight out. So I'm very pleased to be here. And I was asked to speak on probiotics, and particularly on mechanism of action. I'm very happy to talk about mechanism of action because I think that's really the scientific issue when it comes to probiotics. If we can understand mechanism of action, I think it will lead to a lot of opportunities within the field. This is why we need mechanism of action, because there are a lot of products on the market which are claiming to be probiotics and claiming to have probiotic functions, which quite obviously have nothing to do with the kind of science that most of us are involved in. A probiotic floor cleaner, a probiotic aftershave, probiotic mattress covers. It's very hard to know how this links to the kind of science that most of us are doing. So we need mechanisms so that we can dispel this kind of negative publicity, I suppose, surrounding probiotics. In the EU, probiotics have become particularly problematic because in directive issued by the European Union and taken up by Ireland in this case is that the term probiotic now itself constitutes a health claim, and therefore you cannot use the term probiotic on a product unless you have approval from the EU. And they've even extended that in Ireland to the degree that the words live or active imply probiotics, which imply a health claim, so therefore you can't even use the words live or active. Now this is crazy because it's misleading the consumer not to tell them there are live bacteria in a particular product, but we're at a very interesting point, I think, in probiotic science and regulatory approval. But sometimes we present, as I am doing here, almost like the scientists are the good guys, the clinicians are the good guys, and the regulators are the bad guys trying to prevent us from benefiting the consumer. But I think it's much more likely that there is actually a virtuous cycle for probiotics. We all want the same thing. Consumers, on the bottom right there, they want reliable information. They want information that they can trust so they can make decisions on which probiotic they'll buy or whether they'll buy a probiotic or why they should buy a probiotic. And scientists, of course, want to be informed by consumer needs and want to do science of the very highest quality and to make an impact on society. That's why most people get into science in the first place. And industry, of course, are a vital part of the virtuous cycle because they want the quality products with validated and understandable claims. But the regulators are also part of the cycle because they protect the consumer and therefore protect the whole field from misrepresentation, like you get in a probiotic floor cleaner. And the thing that we need, I think, that will complete this circle, the thing that will make this circle possible is mechanisms. If we understand the mechanism, if scientists, clinician scientists can discover mechanisms, then industry can make sure those mechanisms are present in the strains they sell, regulators can regulate it based on those mechanisms of action, and consumers can make decisions. So mechanisms really, I think, is key to completing this circle and getting where we all want to be, which is to have really valuable probiotics on the market. Now, we all know there are lots of different probiotic targets, and I won't list them. Many of them have been mentioned already, and there are many that aren't on this list. I just simply wrote this off the top of my head. If your own favourite target isn't there, I'm sorry, but there are lots of different targets. There are also lots of different probiotics. So what is the link between them? Is it a case that every health target requires a unique and specific probiotic? Is there a probiotic for a particular health target? I think it's pretty obvious that that cannot be the case. There can't just be simply one strain for every particular disease. Are some probiotics effective against multiple targets? Are some targets responsive to multiple probiotics? Are these things counterintuitive? Do they go against each other? Can all these three things be true? Can we have specific probiotics for specific targets, but some probiotics work across targets? I think because we don't have mechanism, it's very difficult again to unravel these questions. Now, there is certainly some specificity in probiotics. This is a very nice paper from Tiffin, Michael Clearbasum's lab, and what they show here was three different probiotics, three commercially available probiotics, all lactobacilli, elicit completely different transcription responses in vivo. These are studies actually done by taking biopsies from the gut of volunteers. And there's some immune genes listed there, or immune genes shown in these heat maps. And what you can see is that the volunteers responded differently to the three different probiotics. So certainly some kind of level of specificity in the way the immune response, the way the immune system sees a probiotic. And interestingly, some of these responses overlap with the transcription responses you expect against certain therapeutics. So it may help us to think that these microbes may well have therapeutic functions. There's evidence of specificity as in things like anti-infection. This is a slide taken from my own lab, from Sinead Carr's work for her thesis. And what we did here was we infected 6 mice, each one representing a group of 5 mice in this case. We first fed them various different probiotic strains. You see 6 different probiotics. And then we infected them with listeria. And we use the listeria that emits light, so we can follow the infection through the body of the living animal. And what you can see is that in 5 of these 6 cases, the probiotic had no protective effect. The listeria infected, the hot spot there of infection is the liver and the gallbladder, but one probiotic did protect, where the other 5 did not. What's the mechanism of action? How can we again choose one probiotic over another? Going into this experiment, we had no idea which one would work and which ones wouldn't. So there is some specificity. But there is also evidence of lack of specificity. And in this case, I'm looking from the disease point of view. This was done, I did this on Wednesday or Thursday this week. I just went on to PubMed. I used Lactobacillus rhamnosus GG as a search term. And in the last year alone, just in the last 12 months, there have been reports of Lactobacillus GG having a positive benefit in all of the conditions you see listed there on the left. Intestinal permeability, non-alcoholic fatty liver disease, lung infection, obsessive compulsive behavior, fussing and crying in preterm infants. So from behavioral challenges to infectious challenges, viral infections to non-alcoholic fatty liver disease, and yet GG shows a protective response in all of these conditions. So what does that say about specificity? Does it say that Lactobacillus GG can do everything, or that there are many conditions out there which respond positively to an influx of harmless commensal bacteria? And the nature of that bacterium doesn't actually matter that much. Remember that Lactobacillus rhamnosus GG was not isolated because it worked in any of these conditions. It was isolated because it gave a big fat colony on a plate. That's why Golden and Gorbach picked it in the first place. Not because it worked in non-alcoholic fatty liver disease, or not because it worked in rotavirus prevention. So why can this strain have all these properties? There's also evidence of lack of specificity when you look at these meta-analyses and systematic reviews, one of which I show here, which used 63 randomly controlled trials, over 11,000 participants, and used a whole variety of strains and blends of strains, and overall what they could conclude was probiotics fairly generically work in the prevention of antibiotic-associated diarrhea. Some worked, some didn't work, but why were these strains chosen, and why were these strains effective, and why were other strains not effective? Is it that antibiotic-associated diarrhea is one of those conditions for which the probiotic doesn't really matter very much, whereas there may be other conditions where the probiotic matters very significantly? A very nice paper appeared in July 2007, which I often refer to. It involves a Danone product, Actimel, and in this study what happened was a couple of clinicians at a hospital in London randomized people coming into the hospital who were going on antibiotics, and they gave them either an Actimel or they gave them a pasteurized milk drink which didn't contain any live organisms. And it's difficult to see the table here, but basically they got a statistically very significant improvement in the outcome in terms of Clostridium difficile associated diarrhea, or diarrhea in general, from the patients that got the probiotic rather than the patients who got the placebo control. Again, when Danone originally chose the strain Lactobacillus casei, they did not choose it on the basis of clinical trials which prove this. The clinical trials came afterwards. So why does this particular probiotic work? It's got a lot of attention at the time, and this is the headline here is from the Sun newspaper in England. The Sun gets everything wrong, but in this case it got something right. So it's nice to see that the Actimel drink stopped the superbug. That was the actual outcome of that study. Now, in the same month, a relative of mine went to hospital in Dublin, and she had a very bad infection. She was put on IV antibiotics, and when I visited her, this was on her bedside table, next to her bed in the hospital. And it didn't come from the shop, and it didn't come from a visitor, it came from the hospital pharmacy. And so when I visited the hospital microbiologist, because Ireland's a very small country, we all know each other, so I went to him and I said, why are you prescribing a probiotic for my relative who is ill? And he said, because I saw that paper last week in the British Journal of Nutrition, and I thought it was very impressive, or it was I forget which journal it was in now, but I thought it was very impressive, and I decided to put my patients, all my patients going antibiotics, are now going on this probiotic. And I, of course, said to him, well, you know that the London study was done with one probiotic, which is Lactobacillus casei, but you're prescribing a different probiotic, Lactobacillus rhamnosus. And why are you doing that? And he just said, because a probiotic is a probiotic. I don't know. He said, you tell me that the rhamnosus isn't even better than the KZI. And I said, I can't, because I don't know the mechanism. And maybe rhamnosus is better than KZI. But we do need the mechanism of action to make an informed choice. And so that's the point of this particular presentation. I'm not going to say anything that any of you don't know before, but I'm going to try and put it in this context of how important mechanistic studies are. And we know, of course, that probiotic mechanisms can work through probably many different mechanisms and probably all at the same time, but three certain targets for probiotics are the microbiota, the host, and the diet. And we could imagine, for example, that probiotics, just like other interventions aimed at the microbiota, like fecal bacteria therapy or prebiotics or antibiotics, aiming at the microbiota could improve human health, but through that medium of the microbiota. We know that probiotics can act directly on the host, either through the immune system or the nervous system or on barrier function, but directly on the physiology of the host. There's plenty of evidence for that. And we know that probiotics can actually act through the diet, not as part of the diet, but actually act on components of the diet and convert the dietary components into bioactive compounds. And I'm going to try and give an example of all three mechanisms of action to emphasize that all three really do exist. So when we think about probiotic mechanisms, we can think that here's the probiotics arriving, usually as part of the diet, of course. They interact with the microbiota, which can be a very dense population of organisms. You've got mucus, you've got the epithelial cells, you've got immune cells. I'm not showing the enteric nervous system. But there's likely to be a mix of specific and widespread mechanisms. It may be that virtually any commensal harmless bacterium has an effect on barrier function. That may not be true. We just don't know because we don't, again, know the mechanisms. We also have to remember, of course, that the gut is a very long organ. And it could well be that the mechanisms could be site-specific. There might be one mechanism acting in the ileum, a different mechanism acting in the colon. There's a different environment. The diet, of course, has essentially been exhausted by the time you get to the colon. There's lots more cells in the colon than there are in the ileum. So we're looking at very different environments. We once fed a 5-strain mix to pigs. And in the 5-strain mix, there were 5 different lactobacilli. And when we looked in the feces, one of those strains was completely dominant. 99% of all the lactobacilli we recovered was one strain. But when we sacrificed the pigs and looked in the ileum, it was one of the other strains that was completely dominant. The one that was in the feces, we couldn't find at all in the ileum. So we've got to remember it's a whole ecological niche, and we shouldn't just talk about the gut. We need to be very specific to the area. So let me give you an example of something acting through the microbiota. I'll use that same example I just showed you a while ago, where one probiotic, this Lactobacillus UCC118, protected the mice against infection where 5 others didn't. And I'm only showing you 5 others. Many others didn't in this particular case. And this was a lethal infection of Listeria. So this was a life or death outcome. When we infected these mice, they died unless they'd got the single probiotic that protected them. So it's a very dramatic readout. Not just light coming out of a mouse, but life or death. And what we predicted, of course, was that there could be a lot of different ways in which an organism could protect against infection. It could act directly against the pathogenic microorganism, for example, by direct antagonism. Or it could act through the immune system. It could be that the probiotic talks to the immune system and causes changes which makes the immune system more resilient against Listeria infection. And Listeria is an infection of the immune system. Or it could be things that we generally term under exclusion, improved barrier function, and thicker mucus layers, effects on the commensal bacteria which lead to protection against Listeria. It could be any of those reasons. We observed that UCC118, the strain we're particularly interested in here, actually produces a bacteriocin, a peptide antibiotic, which kills Listeria monocytogenes. And so this is an obvious potential mechanism by which UCC118 could protect against Listeria. None of the other organisms had that particular property. And when we knocked out that function, so we were able to go in and genetically obliterate the ability to produce the bacteriocin, then there was no protection. So a strain which cannot produce the bacteriocin does not protect strain which can produce the bacteriocin does protect. The ones that just came up there are the ones that don't produce the bacteriocin. And when we look at the light, we can see again, those that get the probiotic in the middle, they're completely no light, they've killed all the listeria. Those on the right are getting the same probiotic with just one gene deleted. So now we're really getting towards mechanism. One gene, one peptide, and one mechanism, a protective effect. However, against Salmonella, a completely different pathogen, this probiotic also protects, but the bacteriocin has no role in this particular protection. So same strain, different target, different mechanisms. This is one of the few examples still where a probiotic effect can be linked to a single gene product. We can also get bacteria acting directly on the host. For example, probiotics in IBS. We know that probiotics work in IBS, at least many different probiotics work, and you see again a meta-analysis here where they say the magnitude of the benefit, the most effective strains and species are uncertain. We went at it in this following way. This is Fergus Shanahan, Naaman Quigley. Rather than starting with a probiotic and testing in the disease, we started with the disease and tried to find a probiotic. So they isolated thousands of bacteria from healthy subjects, brought them down through a kind of a funnel of different tests which made us think they might work in IBS, and ended up with two bacteria that went into clinical trials, a lactobacillus and a bifidobacterium. This is the placebo effect. These are symptom scores you see on the axis. Placebo effect that you always get with IBS. The lactobacillus did not work. The bifidobacteria did work and reduced the symptom scores significantly. So again, specificity here, at least to the genus level of bifidobacteria. This was repeated in England in Peter Warwell's study, and a product has now been launched both in the US and in Ireland based on this particular product. So we know probiotics can work through the immune system. Ted Dinan and John Cryan in the APC have shown that Lactobacillus rhamnosus JB1 can actually change behavior in animals by the production of GABA, gamma aminobutyric acid, which leads to increased levels of GABA receptors in the brain and changed behaviors in the mice. So again, we're down to specific molecules being produced which lead to a very specific effect, which can be measured physiologically. You get increased levels of GABA receptors in the brain, reduced levels of corticosteroid, and altered behavior patterns. Is this a feature of all strains that produce GABA? We also know things can act through the diet. Here's Catherine Stanton and Paul Ross. And what they've shown is if you take strains, this one is a Bifidobacterium breve, which have a particular enzymatic activity, and in this case, they have the enzymatic activity that they can convert linoleic acid to conjugated linoleic acid, which is a bioactive compound in the diet. Then they could change the liver, adipose tissue, and brain tissue, change the CLA content in distal tissues from the gut by simply including this bacterium. If you use a bacterium which cannot convert linoleic acid to CLA, this does not happen. And you can actually then convert a new probiotic into this by, for example, transferring the enzyme from the bifidobacterium to a lactobacillus. And now the lactobacillus can have this impact. So again, the beauty of knowing mechanism means we can take control of that mechanism. We can select for strains with high activity. We can even move that activity to other strains. So we really are getting control over these probiotic mechanisms. So I've shown in a very brief few slides there some examples at least of how probiotics can act through the diet, converting linoleic acid or sunflower oil into CLA or conjugated linoleic acid, and thereby affecting brain tissue, liver tissue, adipose tissue in pigs and in mice. We've also shown how probiotics can produce bacteriocins which can act through the microbiota, because in an infected animal, of course, the infectious agent is part of their microbiota, and shown that probiotics can act on health through producing GABA or interacting with the immune system in very specific ways. So I think the answer to the question is pretty obvious, and it was obvious from the start. Some probiotics are certainly effective against multiple targets. But that may mean they're active through different mechanisms, common mechanisms. Some targets seem to be responsive to multiple probiotics. But I don't think anyone has really shown that a given health target requires an absolute specific probiotic to be actioned or to work in that particular condition. So I think we can divide probiotic mechanisms at least this is theoretical, into things which are likely to be quite rare, strain-specific effects. Doesn't mean only one strain on Earth has it, but it means you have to have the right strain with that particular mechanism, with neurological or endocrinological or production of specific bioactives. We will see other mechanisms which are frequent, which are quite common in a particular genus or species, and therefore you can reliably expect to find in lactobacilli or bifidobacteria. And you find others which are widespread, which virtually any harmless bacterium will confer on a host. And so the suggestion, and this is it's part of the outcome at least from an ISAP consensus panel that was held in earlier this year in London, is that we propose, at least to regulators and to industry and to anyone who's interested, that the word probiotic should not be banned. The word probiotic should be permitted for any product containing live microbes from common probiotic species. And that the body of research supports the fact that such strains can be reasonably expected to have beneficial effects, particularly in healthy individuals. As Johan said, these products are designed for people who are already healthy. They're designed to be prophylactic. But it should not be allowed to have any claim other than contains probiotic without direct and significant clinical evidence to support that claim. So in conclusion, I believe, and I hope most people in this room believe, probiotics are effective interventions in preserving health, in restoring health, and I believe that we're actually at a point where dietary guidelines should include the dietary advice that you should eat high numbers of harmless bacteria every day, in the same way you should eat other nutrients in high amounts every day, whether that's in the form of yogurt or probiotics or fermented pickles or meat or fish. I don't think matters as much as we might think. I think scientists, clinicians, industry, regulators, consumers, we all have the exact same shared goal: to bring excellent science to bear without misrepresentation, without having probiotic floor cleaners. But determining mechanism is essential to that. We will really not be able to advance this field until we get towards mechanisms and understanding mechanisms and how widespread, frequent or rare they are within the species that we study. I'd like to thank my co-conspirators in the Elementary Pharmabiotics Centre who are listed there. I won't go through them by name. Obviously, I'd like to thank the Foundation for inviting me to come here, and you for your attention. Thank you very much.
Thank you, Dr. Hill, for that very lucid presentation. Briefly, he emphasized the importance of studying mechanisms of action, which will help the scientists, the industry, the consumers, and the regulators all to define exactly the role and will help in further promoting the role. He also highlighted the various possible mechanisms related either to the diet, the microbiota, or the host himself. And I think his presentation brought out how little we know still about this. So I would request if you will answer a few questions. Right. We'll take up 3-4 questions, please. Dr. Batish.
Very nice presentation, and this is what exactly I was expecting, that this kind of strategy for selection of a particular probiotic is going to be very useful. But I have a clarification. You said that some are specific for particular characteristics, whereas some strains they are multiple. They demonstrate multiple effects. So it's very difficult to delineate the two kind of situations, because when we feed the strains or the preparation into our gut, it goes into our gut, then we are targeting a particular kind of function, but not necessarily that will be the only one which will be expressed. There will be other functions also. So it's very difficult to delineate the two kind of situations, because it is based on a marker. Are you understanding my?
Yes, I think you're correct, of course, but I think what we will probably find, and this is speculative, but there is some targets, some conditions which involve barrier effects and generalized effects, or deficiencies in generalized effect, will respond to a wide range of bacteria. I think, going back to Johan's talk again, where he showed that we've evolved, bacteria have evolved to ferment milk over 8,000 years, but humans have evolved to live with bacteria for hundreds of thousands of years, if not millions of years, between mammals and bacteria. And I think the immune system has evolved to expect to be exposed to lots of bacteria. And now we live perhaps in a world where we're not exposed to as many bacteria as was formerly the case. We don't use fermentation as much as we used to use in the past. And so I think the immune system responds positively to seeing high numbers of bacteria, almost regardless of the bacterium. But then in other cases, we will have bacteria which have specific enzyme activities, or they produce a particular bactericin or bioactive, where obviously it is crucial that if you are addressing a particular health state, you must have a strain that has that particular mechanism. So I think that the divide is fairly straightforward.
No, do you advise that we should use this multiple kind of strategy or a single function-based strategy? This should be for screening purpose, because it's very difficult. We are finding lots of problem to find out the correct strain.
Yeah, I think, again, I think it'll be disease condition specific. I think in certain diseases we could use any of the existing probiotics and they're just as likely to work as going to look for a new one. There would be other conditions where we don't have the appropriate probiotics in the arsenal already, and screening strategies would be the only way to get these new bacteria.
May I request the other questions, please? Yes, sir.
There is one small
utilize
question
the best time.
that how do the European regulators see in the wide spread functions, which are generalized function, as you say, barrier function, building of immune systems, etc. So these are with most probiotics, these are there. So do regulators allow European Union to write probiotics for widespread functions?
Yeah, I think the regulators, they have a bad reputation among scientists, I guess, because they seem to always go further than we think they need to go. But I think in this case that's fairly well deserved. I think they have gone beyond their remit, really. Their remit is to protect the consumer from misleading information. And I think protecting the consumer from a claim which is not validated in clinical trials is reasonable. To protect the consumer from the information that their product contains live, harmless bacteria, that's not correct. They're misleading the consumer in that situation, I would suggest. And I think it's important that we lobby the European regulators to accept that point of view, and ISAP is going to try and do part of that at least.
Okay. You're going to have only two? Yeah. Sorry. Yes, lady first.
Thank you. I just wanted to find out, you were talking about the GABA producing bacteria. Now GABA is also produced by human tissues. So do you think the supplement, if we supplement with GABA producing bacteria, it will give a better benefit?
Well, in the case, again, it depends. I mean, what Ted and John showed in the study that I showed was that mice fed a GABA-producing bacterium responded by producing more GABA receptors and by changing their levels of various different cytokines and also corticosterone, and they behave differently. But these were mice that are in captivity, they're anxious animals anyway, and therefore you're relieving that anxiety to a degree.
Okay.
But that's a kind of a very artificial situation. Would a wild mouse living completely anxiety-free benefit as well? I'm not sure.
I have also come across one study where they have said Bifidobacterium dentium produces a profuse amount of GABA.
Yes?
And so that's what I was just wondering, is there a crosstalk between the ENS and the GABA producing?
I think it depends, again, on where the GABA is produced, because a lot of the bacteria in the gut, a lot of the proteobacteria produce a lot of GABA. And if you eat glutamate, E. coli can convert it into GABA. So GABA is not a strange molecule to find in the gut. It's just the level at which you add the bifidobacteria probably make a difference.
Okay, thank you.
One quick question.
Last question.
Yeah. Is there a role of the dose of microbiome in telling us the efficacy of the probiotic. For example, these many colony forming unit, these many billion colony forming unit also guide us or depict the efficacy of the probiotics.
Yes.
Which I didn't see in your slides.
Yes, I mean, if it's real, there must be a dose response. These are not colonizing organisms, so you can't consume them at low dose and then they just simply grow up to the required dose. You must consume them in adequate amounts. And I think for most of the functions that we're talking about, adequate amounts is very high relative amounts of 10 to the 9, 10 to the 10, I think.
Okay. Is there any fixed dose that has been recommended or anything you can think of?
Not yet.
Okay.
No, because there's so many different probiotics, but I think 10 to the 9 is a reasonable figure to aim for.
Sure.
Okay. I think we cannot permit any more unless, as a chairperson, sir, you can ask.
It's quite intriguing that the dominant bacterium in the feces was not the dominant one inside the gut.
The ileum, yeah.
And then in which case, now if you go by this as a marker of the dominance you estimate, it will be going absolutely
Yeah.
wrong.
Yeah, in that particular case, it was an interesting one. We had 5 bacteria, as I said. One of them produced a bacteriocin which could kill the other 4. So we thought the one that could produce the bacteriocin would dominate. But in the feces, another one dominated. But in the gut, the one that could kill the other 4 dominated. So again, we're looking at completely different ecological niches throughout the gut. And we're using feces very often as a reporter of what's happening 20 feet further back up the gut. It may not be a good report.
Well then, thank you very much, Dr. Hill, and we come to a conclusion for this session.
Thank you, Professor Hill. That was an excellent talk. Thank you, Professor Mittal and Dr. Shashikaran. A small token of our appreciation for Professor Hill. Can I request Professor Mittal to kindly give a small token? Can I request Mr. Minoru Shimada and Mr. Tomoyuki Iwama to kindly give a token of our appreciation to Professor Mittal and Dr. Shashikaran? Mr. Minoru Shimada, Professor Mittal, please. I think we'll open the shawl and make him wear it. Mr. Iwama for Dr. Shashikaran. Thank you, Chairpersons and Professor Hill, for an excellent insight into the mechanism of action of probiotics. And we move on to the next session. I now request Professor Anand Krishnan and Dr. Uma Chandramouli to chair the session, Epidemiological Studies on Probiotics. Professor Anand Krishnan is the Professor, Center for Community Medicine and Head WHO Collaborating Center for Capacity Development and Research in Community-Based NCD Prevention and Control, All India Institute of Medical Sciences, New Delhi. He is a Fellow of Indian Public Health Association and Indian Association of Preventive and Social Medicine. He is a recipient of BC Srivastav Award and MK Shishadri Award. He has more than 150 publications in peer-reviewed journals. Dr. Uma Chandramouli is Assistant Professor, Pediatric Biology Center, Translational Health Science and Technology Institute, Gurgaon. He has been awarded the Ramalinga Swamy Fellowship from the Pediatric Biology Center, THSTI, and is a recipient of several awards, including the P.N. Taneja Award. He has several publications in peer-reviewed journals and 2 book chapters. Can I request the chairpersons to kindly introduce the speakers, Professor Sunil Sazawal and Dr. Radhanath Satpathy.
Professor Sunil Sazawal is currently an associate professor at the Bloomberg School of Public Health, Johns Hopkins University, Baltimore, and is the director for the Center for Micronutrient Research, Clinical Trials and Operational Research in Maternal and Child Health, New Delhi. He has received the Rockefeller Foundation Special Postdoctoral Fellowship in Clinical Epidemiology and Advanced Studies, and has several awards and publications, nearly 100 of them. He's the first speaker for this particular session.
Put this in. Honey, ये इसके पॉइंटर के लिए मैं ये यूज करूँगा, उसे स्लाइड पे घूमूंगी. है ठीक है good afternoon. It's when you stand up on a podium this late in the afternoon, I always remember an anecdote from Indian history that a gentleman went to a war and then he got the arm of the soldier, and then somebody asked him that, I thought the norms of the war are that you're supposed to get the head. He said, somebody else got to the head before me. So I guess enough has been said about it already, sir. But we'll try to change our stance. I think that we could. I'm trying to figure out this guy first. फॉरवर्ड कौन सा है नहीं चल रहा है No. Okay. All right. Sorry about that. So I think the talk that preceded was actually a really kind of setting up the stage for this. And while I was listening to Professor, I remembered our days with zinc. We had the same dilemma. Zinc can impact 230 enzymes in the body. And then if you are really trying to understand which of those is the mechanistic possibility of a particular action, it becomes difficult. Which doesn't mean that we shouldn't try it, but it becomes difficult. And I think that was part of the talk that we had, is that there are so many layers of mechanism of action that either you need to work on each one of those, or you need to kind of do that in a very systematic fashion. But before you do that, that's where I guess some of us come in. Our intent or our idea is that ultimately we are interested in trying to see what works. And in fact, my mentors, Dr. Ban and Dr. Black, we had this discussion that what works is what is proven in a double-blind randomized controlled trial. That's the only way to assess if what you are saying, it actually is happening. And so one of the ways of kind of moving forward in the area of probiotics is to say, all right, what is the evidence that we have so far from randomized controlled trials in terms of impact? And I would limit, because like a gentleman rightly pointed out, if you try to do Google, you would have 5,000 hits, and some of them will not be randomized trials, some of them will be varied kind of symptoms, and I don't want to get into all the symptoms, so we're trying to focus on what do we know at the moment about role of probiotics in childhood diseases. And that's what I will try to present two sides of the story. I'll present some of the evidence that we have tried. We did a meta-analysis some years back, but I tried to update it with the data that is available as of now, that what do we know about impact on childhood morbidities. And then also present to you, because unfortunately there are not that many studies that look into other morbidities in children, and I think our study still remains one of the only ones done in the community setting, which we will come to that, because there is a lacuna in that field. So I will present some data from there to give you a feel of what we found from that study. And before I kind of get into this, I think there was a discussion, and I would start with that there is this need of defining a probiotic. Like we understood that there can be different mechanisms, But unfortunately, there is a problem. I mean, there is a problem on the other side. I mean, there is a problem that you can go to the regulatory bodies and say, you know, we can go and claim that everything that has a live organism in it is a probiotic. But then the trouble is that then people take it to the next step and assume that because it is a probiotic, it has all the activities and efficacies of a probiotic that is, or any other probiotic that is up in the market. And that needs to be kept in mind. And this area needs to develop in that direction, that we need to have better definitions of that. So, I mean, when I am talking of the impacts of probiotic, I do not want to be quoted tomorrow as saying that any probiotic has these effects. It obviously belongs to whichever ones have been tested. Whatever is there is basically based on that strain, or based on that dosage, or based on that trial. All right. I'll skip through this very quickly. We know that diarrhea and, the idea is that diarrhea and pneumonia and acute ear infections are important causes of morbidity, and I don't know to tell you that. And there is accumulating data that we have heard today, and from studies which suggest there could be a benefit using probiotics in a multiple symptoms or syndromes, sort of. So I already told you that I'll try to give you like a meta-analysis in the under 5 children of what we know about morbidity, especially these important morbidities related to probiotic supplementation. The issue that I want to highlight, and I'll highlight it during my talk as well, is that this area, there has been an extrapolation across the board. You know, you seem to feel that the impact on antibiotic-associated diarrhea is the same as, diarrhea in a developing country, is the same as a hospitalized child, is the same as a daycare setting, and that needs to be kept in mind, that those are not the same things. And that's why, although there is a lot of data, we need to bifurcate the data and see how does it pan in terms of different conditions, and how does it then pan in terms of the conditions that are basically important to us, depending on where you are. I mean, if you are in a hospital setting, you're treating very, sick children who need very high doses of antibiotic, it's a different condition. If you're working in a community setting and you're trying to prevent diarrhea, you're working in a different setting. So that paradigm, I would like all of us to keep in mind. Okay, so this is the data that we have from what we tried to do was, first of all, as a first cut information, try to get information on all randomized controlled trials where we felt the methodology was adequate, which were well done, and then tried to do their impact assessment and a meta-analysis. And as you can look at this graph, this is standard meta-analysis where each study, the size of the box gives you how much of a weightage it has depending on the number. This gives you the impact it had, this gives you the confidence interval, and then the bottom here gives you the summary impact that is based on these studies. Whenever I discuss this graph, I always try to emphasize on people that do not only look at this, look at all of this. So you need to look at, what do we see here? We see that most of the studies that are there have shown a beneficial impact. Most of the studies are on one side of the graph. And the other thing that you see, which is very important that I want you to look at, is the larger two studies. This was our study in India, and this was Abraham in Peru. And these were the only two studies that were actually done in a community setting. They were larger studies, and they were done in a community setting, and you see a smaller effect in those studies. That's all that you see, but overall, if you pool the data, you are seeing a significant impact on diarrhea. Now, this gives you the details of the studies, and I won't bore you with that. at the moment, but it's the same thing that it gives you, whether it was an active or a passive surveillance, and that would impact some of the quality assessments. And given the time today, I'm not going to go through all of that in detail, but it is here for you to look at it. And again, like I said, these are the only two studies that are done at the community level. Rest of the studies were basically at health facility level, either at a hospital or at a health facility. Now, what did we try to do? We tried to then say, okay, can we separate it down by antibiotic-associated diarrhea? Because that was, all the facility studies are basically focusing on antibiotic-associated diarrhea. So, this gives you now comparison by antibiotic-associated diarrhea versus others. So, you do see that there is a slightly higher benefit that occurs across the board with antibiotic-associated diarrhea, and if it's not an antibiotic-associated diarrhea, the impact is slightly lesser. That's the bottom line message. Now, the other thing that we have been discussing today, and that was the question we were asking at that point, is it, how specific is this impact based on a particular strain? Is it only, algae has an issue? Is it that there are, the trouble there is, respecting the data that was presented today in terms of laboratory investigations, again, these are small studies. The fact of the matter is that the data about is not sufficient out there to really make a comparison across the strains. This is what you see, is that there are 4 or 5 studies with algae, there are 3 studies with this, then again as you go down to Bonita, there are 2 studies there. So although it doesn't, at least from this analysis, it doesn't seem that there are drastic differences, at least in terms of diarrhea, among the strains, among different strains, as of now, as the literature is now. But one needs to keep in mind that there is a dearth of literature in this area, and there is need for more studies to really look at if there are across the strain effects, and is there a reason for choosing one over the other for diarrhea. At the moment, that evidence doesn't seem to be available. Then if you look at the other idea has been that most of the studies have been in developed countries. And again, those are either at the facilities or they are at, and as we know, the gut in both of these settings is totally different. The colonization of gut is different, how the gut responds to bacteria is different, what is normal exposure of gut is different. Then added to that, you have malnutrition where the mucosa is different. So given that, what we tried to do was to separate it out into developing country studies and developed country studies. And again, you see 2 things there. You see that there are very less studies. You got 5 studies in developing countries. You got 2 of them which are community-based studies which did not show a huge impact, which have the major kind of a, sort of weightage given the sample size of that. And then you have got 4 studies which are very small sizes and which are some of them broad kind of confidence intervals. Overall, even if you bought and said we still want to go ahead and look at the meta-analysis, you still see the same thing, that the impact that has been shown classically in the developed countries, the impact in the developing countries as so far has been little lesser. Now, but the caveat, we need all to keep in mind that there is lack of data, lack of real data from double-blind, randomized clinical trials at community level of adequate sample size with adequate outcomes. It isn't there. Then we were looking at, does it matter where the children are kind of enrolled, and as you can imagine, it does. But the trouble there again is that you got majority of the studies from hospital settings. That's the trouble. The reason that I presented these slides to you is that when you see a meta-analysis and you see all these 20 slides, lined up there, you don't get a feel where are these data coming from. So you assume, a clinician in a field will assume that, what this tells me is that if a patient walks into my clinic tomorrow, that's the same. But that's not happening. And the thing that you again see, which I highlighted, is that there are only 2 community studies. I was actually told that there is probably one more community-based study available now from India. And so, but that's all we have. We do not have community-based randomized controlled trials available evaluating the impact. All right. So I will not go into this. You already know we try to look at the subclasses of the strains and all of them, but I don't think, you get the message, and so I'm not going into the details at this point in time for the sake of time. I will also not, I'm flashing this, but just to say that we did go through the whole process of doing the agar plots and doing publication bias issues, and then compensating for publication bias, and then looking at if that is impacting the results and compensating the results for that. Just to say that it was done in a methodologically sound way. The other thing I wanted to share with you is the recent publication which has come on, on pneumonia, on respiratory tract infections, and these are the data from that analysis. Again, marginally small, but overall impact on areas, less number of studies. If you look at otitis media, seems to be little more impact on otitis media than on overall pneumonia. Again, antibiotic requirement for antibiotic treatments, overall some significant reduction, but studies are very few. So I guess I have, 3 minutes left to speak? Okay. So I will in 3 minutes try to, the idea was to get you that we did a study which, where we looked at a double-blind randomized trial of pre and probiotic given as a milk, with the milk base. So I will essentially, I don't think I'll skip the design. It's a double-blind randomized controlled trial at a community-based setting, involved about 624 children, 300 children per group, as you can see. And the other thing that you need to see is that both groups actually got same macronutrients. The only difference actually between the groups was, including zinc and iron, after all, and the only difference was the probiotic between the groups. So whatever difference you're seeing is that it was not only that they were getting normal diet within the milk, both the groups were getting a supplemented milk, so to say, within higher quantities of milk and iron. So this can go further, and there are 3 things that we found there, which in addition to diarrhea. First thing that was there was that the diarrhea impact, I won't go to baseline, they were comparable for everything that we tried to look at. And compliance, first was the compliance was very good, but 90% compliance in everybody. So, it was much better than we have ever had with any drug or iron or, in the community. So the other thing important was that we did see, which is not reported elsewhere, and that's why I wanted to highlight this, is that we did see, as I said, a low effect, about 8%, 9% effect on diarrhea morbidity. But in addition to diarrhea morbidity, we did see, because we were doing a constant morbidity check, there was an impact on days with fever. So there was about 5% reduction in days with fever. There was a significant decrease in days with high fever. There was a significant difference in days with severe illness, which was not only diarrheal illness, which was any illness, which included febrile illness, maybe a viral illness, where a physician basically said the child was severely sick, so we had a score there while it was sick. And there was a significant decrease in measles. the numbers are very low. I mean, so it was 5 versus 10. So we should not interpret it as 50% reduction. I wouldn't do that as an epidemiologist. But there was a reduction in measles illness. Okay. So what do I need to do? All right. Give me a minute to wind up. So that, I mean, the reason that I wanted to share, again, if you go into the diarrheal episodes, there was a decline in dysentery episodes more than simple diarrhea episodes. And days with bloody diarrhea was lower, and if you see, the need for antibiotic was lower. So obviously, what I'll not show you now that there's no time is that there was an impact on our hemoglobin. Now, this all could not have been directly responsible. Either it was responsible with the immune impacts that we have discussed in terms of the mechanism of probiotics, as well as better nutrient absorption resulting from an improvement in the gut. So it was not only a question of impact on diarrhea, which was there, but there was an impact. I think that's where I'll end. There was some effect on growth, which is again consistent. We did have an improvement in weight velocity, and again, the consistency is the same. They were getting the same diet, but there was an improvement, indicates that there was an improvement in the gut functionality that ultimately resulted in it. Thank you.
The next speaker is Dr. Radhanath Satpathy. He's a pediatrician who is the head of the Social Pediatrics and Minority Health at Asian Institute of Health, IGH AIPH Center for Translational Research at the ISPAT General Hospital, Rourkela, India. He's done his fellowship in pediatrics through an Indian Academy of Pediatrics fellowship at AIIMS, and he has more than 30 publications in national and international peer-reviewed journals.
Honorable Chairpersons, esteemed delegates, I must be thankful to the organizers for giving us the opportunity to share our finding of this study. This was a field trial of Lactobacillus plantarum, symbiotic therapy for preventing late onset sepsis in neonates. This was funded by NICHD, and I am Dr. Satpathy as a co-author presenting this study. A lot of things have already been discussed about the use and the need of probiotic for adults in clinical practice, in experiments. We basically consider as clinicians the appeal for probiotic in neonatology is threefold. It is an attractive option to more aggressive neonatal therapy, therapeutic interventions. It is a simple attempt to recreate normal natural flora, gut flora in neonates, and this is mainly used for disease prevention. And as we know that we have already discussed several times that for different diseases, different kind of probiotic, for different organ, different type of probiotics. But broadly, I must say that as a clinician, and probably finally when the matter of delivery of probiotics to the children comes, we consider these are the food substances mainly used for disease prevention. Our background is most probiotic trials to date have been formed using preparation available in the market. Diverse strains and strain combinations have been used. Some strains have been tried in multiple disease states with uniquely different pathogens. We have discussed it today. Colonization of the strain has been seldom studied, if ever, has been examined in this trial. Results have been extremely varied. These are the types of probiotics available in most of the Indian market. And this is a baby with neonatal sepsis. The preliminary studies suggest that this study was designed after extensive scientific investigation. Surveillance of neonatal sepsis in India demonstrated that bacterial translocation from the infant gut to be an important factor in the pathogenesis of sepsis that we have discussed today. And multiple hospital-based clinical trials were done to examine colonization in neonatal gut and safety. The current preparation we have used is Lactobacillus plantarum, remained colonized in the infant gut over 120 days after 1 week of oral dosing as demonstrated by stool examination. These are the earlier publications of our team. Then our study design was basically it was a individually randomized placebo-controlled double-blind clinical trial in the community setting in India. The study was designed with 80% power to detect a 20% relative rate of reduction in the incidence rate of late onset sepsis. And the Data Management Safety Board stopped the study for efficacy in one of the groups at the interim analysis. We have taken the approval of UNMC and Indian IRB. We have registered the clinical trial. This was conducted under ICMR India oversight with the Health Ministry Screening and Committee of the Government. This is Odisha, where the study was conducted. These are the two places, Sundargarh and Khurda. These are the two places constituting 4 revenue blocks of Odisha. The study was conducted. This is regarding the Khurda district where Bhubaneswar is located, and this is regarding the Ispat General Hospital where I have been placed. This is in Sundargarh district. These are the 2 blocks where we have where we have used the product. Then the study design was individually randomized. 7 oral doses of symbiotic preparation, probiotic and prebiotic administered orally to neonates. Study initiation done above 24 hours and less than 96 hours when infant breastfeeding has been started. Infants were followed daily for 60 days at home by trained field workers. All the suspected sepsis cases and others, adverse events and severe adverse events were managed by study physician at attached hospital. Bacterial blood culture and antimicrobial identification was done at the attached laboratory. In fact, this was a very hectic task, and organizing the field with a population of nearly 2.5 lakhs with around 4,000 births in a year, and educating the mother, educating the recruited worker, educating the ladies was very difficult. But when we started the mission, it was just done in a mission, because where we worked in Odisha is an area with high infant mortality rate. Though I have worked in a very corporate hospital, probably first of its kind in India, still Authority of India has first started corporate hospital in the country as early as 1962. But even then, after working for 30 years, I thought that we have not been able to prevent the child death in the hospital. Therefore, we wanted that we should probably work for community, and when we had the opportunity through this project that we must work for the community, we continued the project. Simultaneously, we have offered care to a very densely populated tribal area constituting near about 2.5 lakhs population, where the women literacy rate is as less as around 25 to 30%. This is how the study team was designed: site director and principal investigator, study physician, and then all these Anganwadi centers. There are near about 150 Anganwadi centers, and all the Anganwadi workers, the helpers, and the newly recruited women, all were educated with intense difficulty, and then they were taught how to administer the probiotic to the newborn babies after taking a proper history of breastfeeding, that whether breastfeeding has been initiated or not. This was personally, the training was imparted by me, it was designed by me also. Then these are some of the pictures where, if you can look into the left bottom picture, this is how we have, in process of dance drama, we have demonstrated in the music that how sepsis can be prevented in newborn babies. And the above top picture is a picture of training, how we are training the Anganwadi workers, and this is one of the study physician who is imparting the training. These two lower pictures are how we are educating the at different places in the rural area to women that how to prevent neonatal sepsis, being aware at a right time. Then this is another one of my colleague who is examining a baby. He is also a pediatrician, and these are the Anganwadi workers who have been brought to hospital periodically and are shown how a septic baby looks like. This is the product that we have used, symbiotic preparation, Lactobacillus plantarum, and the placebo contained only maltodextrin, dissolved in 2 ml of dextrosaline solution and orally administered to the infant. This is the picture of administration of the product to the newborn baby. Then these are the exclusion criteria we have decided for the babies. Again, to continue the exclusion criteria, if the baby was on antibiotic, we didn't recruit those babies. Then these are the number of babies we have screened, around 7,092 babies we have screened, and we have enrolled around 4,556 babies. 26 consent was refused, and so on. This is Dr. Panigrahi, one of our principal investigator who is in the field to discuss with the mother, and the community worker is directly supervising some of our data collection in the field. Then our demography is like this, mean birth, then the weight at enrollment, breastfeeding status, mother's age and mother's parity. The total number of doses administered were this. This is our microbiology team. Dr. Panda is one of my colleagues, and Dr. Judy Johnson from USA. This is our result mostly in the placebo and placebo group and in the symbiotic group. This is a situation where how a Anganwadi worker is preparing the probiotic preparation in the field in a sterile manner, is about to administer the same to the babies. Continuing result, our culture negative systemic sepsis, culture positive systemic sepsis, and gram positive and gram negative sepsis was like this. These are the other conditions that we have seen during the trial. This is our biobanking system available with one of my microbiology colleagues. These are the various conditions as we see in placebo and symbiotic group. They vary with statistical significance. This is a picture of the, this result shows the weight gain in different age group where the placebo and symbiotic group, they differ. The timing of blood culture has been summarized like this. So in conclusion, what we say is the Lactobacillus plantarum symbiotic significantly reduces the incidence of clinical sepsis and culture-positive sepsis in healthy units in India. The symbiotic preparation also significantly reduces the LRTI pneumonias. Apart from the blocking bacterial translocation, the preparation seems to have other immunomodulatory effect on which we have deliberated throughout the day. Early administration and long-term colonization of the lactobacillus plantarum may be critical for the observed effect, but it is very difficult to prove. And finally, this is where we have been checking in the data collection in the rural area going from hospital. And this is the last picture that we have seen. These are the babies who are from the field. They have all taken probiotics during their early childhood. They are still smiling. When we go to the periphery, they seem to be healthy, but as there are many studies, its total impact has to be evaluated, for which we are here. Thank you.
At the outset, you want to say something, Major? At the outset, let me be frank. I am actually a newcomer to this area of probiotics. I have no knowledge of probiotics except perhaps having my dose of probiotics through curd rice, idli and dosa. I hope to therefore learn from this symposium, and I think I was not disappointed. We had a lot of excellent presentations, and I see that a lot of research is happening here from lab to clinical. And now we are seeing that things are moving into the area of community and public health, which is a very good way of moving, moving from lab to patient, bedside to patient to community. We had two excellent presentations. I think both of them answered some questions, but I think raised many more questions, and I'm sure we will have a lot of question answer on them. Interestingly, both of them focused on childhood infections, perhaps one of the most important public health problem facing the country. And we had an original trial, which was a very large trial. About 7,000 children were enrolled. 4,000 were enrolled after screening 7,000, which is really a huge trial. And also we looked at a slightly higher level of evidence, which is a meta-analysis, and we had some, as Dr. Sazawal said, mixed sort of results. Some of them are showing good evidence, but what would be of interest to us, studies in developing countries, community-based studies, normal diarrhea, not antibiotic-associated diarrhea, the evidence seems to be not that sound for us to move towards public health interventions. And he concluded by saying that we need more studies, which I think Dr. Satpathy clearly showed that we are going at it, doing those kind of big studies. And I'm sure some more studies would be done in the future before we are in a position to synthesize that evidence to move towards some action. But now I think the floor is open for the group to take questions. Yes, Dr. Ganguly.
Health effect on the growth was quite significant, and I think this makes us a lesson, and the strains used were different. In one case it was plantarum, in another case this was Bifidobacterium lactis. So when we really look at probiotics in an overall context, we need to have parameters and endpoints which are of general health benefits, which are at least undisputable. If you go across the meta-analysis, most of those papers I've read, this goes across the board. One point I want to emphasize here, which we faced from a person, activist. In Dr. Satpathy's case, they went to the court saying that these were used in infants and there was a risk. What was the safety? Of course, the court case was won, but we had to answer safety issues, and particularly some issues of translocation and other were brought up. So I'm very happy that at least there is a court case in India where a decision has been given about the safety of a particular probiotic in infants.
I think we'll take all the questions and then ask the panelists to, in order to save time. Yeah, maybe lady first.
Yeah, I'm Dr. Jagmeet Madan. I'm a nutritionist and a professor from SVT College of Home Science, SNDT Women's University, Mumbai. I have a question to Professor Saswal. Looking at the study, you said that, the nutrient content of the two groups was identical, and the only difference was the drink which was given containing pro and prebiotic supplement. My question is that when we are looking at the diet, today in our diet also that there are many prebiotic containing foods which are present. So have we looked at the prebiotic content of the foods which were there in the diet as well, besides the prebiotic which was given along with probiotic? Because the probiotic would not necessarily only interact with the prebiotic which is being given as a supplement, but also the prebiotic content which is present in the inherent diet of the individuals. And today, of course, we have a lot of data in terms of the prebiotic foods, naturally occurring foods, which are existing more so in our Indian dietaries. Would you like to comment?
Maybe, I think if we can, I think we'll save time. Dr. Gopalan.
Yeah, my question again is to Dr. Sazawal. Why did you choose a probiotic with a prebiotic? Why not a probiotic alone? And if you had chosen only a probiotic rather than a combination of a pro and a prebiotic, do you feel your results would have been any different?
Anybody else wants a question? Okay, that's the last question that we'll have from them.
I just wanted to add to Dr. Saswal's presentation that we did a study in Kolkata on 4,000 children under 5 years with the probiotic Lactobacillus casei strain Shirota. And we found a 14% reduction in diarrheal incidence between the two groups. And of course, it was a double-blind placebo-controlled study.
Okay, just to balance, because all questions seems to be aimed at Dr. Saswal. One question for Dr. Satpathy from me. Perhaps the most key public health message to mother is give exclusive breastfeeding for first 6 months. That's what we are saying, and we are yet to achieve the success in it. But, and we know in India there is a practice of giving ghutti or barley water, and we are fighting to stop that. But now are we going to say that don't do that but give a probiotic, or are we talking at cross purposes between exclusive breastfeeding and using probiotic as an intervention in the neonatal age group?
We have taken adequate care to see that breastfeeding is initiated in time and continued. When we administer a dose of probiotic during breastfeeding, I don't think that we are trying to give a message that we are stopping breastfeeding. We are giving probiotic with a purpose of doing good to the babies, over which there are significant clinical materials are available, significant literature review has been done to understand the better effect of probiotic in babies. But a single dose on a day, particularly if given along with 2 ml of saline, is probably not going to stop breastfeeding in any case. So that's my personal feeling.
Are we diluting our message on exclusive breastfeeding?
Because we are doing that. I also worked as a promoter.
there is a possibility that one could. Is it okay? There is a possibility, I mean, in their case, I think they tried to give it with saline, but there is a possibility that you could give it with breast milk, right? You could give the same thing with the breast milk as well, expelled breast milk.
That can be also given with breast, but...
So if there is if you want to, you're right. I mean, the question, the moment, if we say that we will give it with water, we will be contradicting the message we are trying to give of exclusive breastfeeding. So, but if we say you can expel the breast milk and then add this to the breast milk and then feed the breast milk, that might be the, programmatic route to move. Anyways, I'll go back to the questions.
I can use the mic maybe.
I just want to clarify that before this probiotic trial started in community, we did studies in the hospital with the same probiotics, and it was a preliminary study which was done, and that time we have used EBM, express breast milk, as the vehicle. That time, we didn't add saline, and we got good result, and from that preliminary, ultimately the community trial started. But in community, they had, certain limitations, so to make a uniformity, that was the procedure which was followed.
One can understand that in a setup, in a trial, probably there are difficulties of implementing something, but the question which was here, which is very pertinent, that if we go forward and say, you can give 5 ml of fluid, then you're changing the message that you can give good tea as well, then. I mean, that's also a medication, so we might change that by giving that message.
One last question from the back there, and then we'll summarize.
Sunil, this is, I think, taking it back to the zinc question that you raised. I think as pediatricians, we've struggled with this whole thing about zinc in prevention of diarrhea, and eventually seeing the benefits of zinc on diarrhea, the practical implications and the application was that look at it as a therapeutic agent. And unfortunately, the probiotic story has just gone on, and I mean, people who've been part of these seminars earlier, we've talked about what you presented earlier, and again it gets reiterated that we just don't have enough evidence. So is it time that we now start looking at more focused questions in this whole thing? Because, I mean, telling a country that you're going to give a bottle of probiotic every day to prevent diarrhea is something which I'm sure many of us are not going to be able to see it as a practical solution. So I don't know whether we really need to now move on to looking at small focus groups as for prevention or therapeutics in future research studies.
Okay. All right. I'll try to go systematically. I think the first question, I think Dr. Ganguly brought up a comment which was more related to the fact that, yes, we are seeing benefits, and that was the point that well, first of all, we need to appreciate that the two trials were in two different populations. So you're talking of, older children, 1 to 3 years old, and then you're talking of neonates, and so the conditions are different, the issues are different. But you're right, you're absolutely right that at the end of the day, ultimately we need trials that are looking at practical outcomes. Rather than saying, you reduce, you have been part of those discussions, you say, okay, we give something and we reduce diarrhea by 1 day, or, half a day. What does that mean at the end of the day? Are we willing to invest for doing that? And so we do need studies, and that was the primary thing that we have faced. And I would, combining the question that I was in fact told today about the Calcutta study, and I would like to get the publication and data from that, which would help greatly, but that's all we have. You have 2 studies in India and 1 study in Peru. That's all you have for probiotic trials, double-blind randomized trials at the community level in pediatric age group. That's your evidence today. And you're making all decisions, whatever decisions you make, you're basing your decisions on that data. it's not fair. I mean, even if it is my data, I would say one trial is not sufficient. I mean, it's not the end all. I mean, you need more data, you need larger studies, you need those age groups in those settings. But what is happening is everybody is quoting data from child centers, quoting data from hospitals, quoting data from 30 children studies in the developed countries with antibiotics, somebody admitted in a cancer ward with getting antibiotics every day and then given probiotics. Is that relevant to a common child in the community? It's not. Anyway, so I totally accept your point, sir. Now, the second point that was raised about the question of prebiotic and I think I rushed through the study, and so it was not very, very clear. What we did was both groups were getting milk. So one milk had all the nutrients I showed you on the two sides. So it was a high-end milk which was fortified with iron and zinc and had other stuff in it. And the other end had the same and had pre and postbiotic. It was a double-blind randomized trial, unlikely that there would be differences and it was a large trial, so it was like, 400 children per group. Unlikely that there were differences. We do have dietary data on them, and we have all of that data. And so that didn't happen. I can tell you that. So it wasn't a question that there was a difference in prebiotic in the food, which then added to the impact. We know that. Now the third question was, why did we select prebiotic and postbiotic, probiotic, and why not only probiotic? Was again that it was our decision at that point was to undertake a practical trial and see what is the best benefit we can get. Because if you would do a probiotic and you don't get an impact, then people will say, oh no, these children are not taking enough diet, there is no prebiotics in their diet, and that is why probiotic is not happening. So you wanted to give yourself the best potential possibility of an impact, and that's why we designed it that way.
The other thing is probably because if prebiotic is administered, and then the possibility of survival of probiotic in the gut probably increases. That's the reason the combination has been decided to be taken in the clinical trial.
You see, the scientific part of it is that the potential, because colonization is an important component, and it's not simply taking bacteria. And we did, I didn't present that data, but we did go ahead and do the colonization data and showed that the same bacteria are actually colonizing in the gut. Because for certain impacts, not for all of the impacts of probiotic, but for certain impacts, a lot number of times people forget that the colonization of the gut by that particular strain, in fact traditionally that was the definition of probiotic, was that if you took that bug, it had to colonize in order to be determined as a probiotic. I mean, it can be live and just pass through the gut. And that, we know from, microbiota studies that the chances of that happening are the best in the age group where they gave the probiotic. The colonization is maximum at that point. As you go further in 1 year time, the colonization will fall down, and anything you can do to improve that colonization is good to do, I guess. So I think the only question finally is Shinjini's question, and Shinji, I totally agree that we need to move forward, and we need to have, that you are aware of the zinc story. I mean, we did studies and studies and studies and demonstrated everything, but at the end of the day, the only thing that has so far gone into the program is the diarrhea treatment, because it is not practical to have tablet every day or liquid every day or something of that kind. So there is, that's the pitch I'm making, is if we have to move the probiotic field forwards, either we have to move it in two terms, that we are saying that there is specific conditions where we are giving it as a therapeutic adjuvant, and then studies need to be done for that. Then you need to do studies to look at therapeutic adjuvant impact of probiotic in certain conditions. If we are saying that we want to have a preventive modus operandi of it, then we have to move towards how. Can we, I mean, I can tell you off the record that I have, one of the colleagues I have in, GB, institute, his point is that he could make a regular curd that can be made in house, which will have all the qualities. So, are we over kind of exaggerating and saying this is something that can only be in a tablet? I mean, if you tomorrow said I can give you a starter, and you make the curd in your house which will have the same qualities, then the problem is solved, right? But nobody has investigated this.
Somebody who asked, why is a prebiotic taken? Okay. Why is a prebiotic taken? We have done the studies wherein we have proved by in vitro cell culture studies the adherence factor at least is proven beyond doubt. It's better when it is given with a prebiotic. When we take bifidobacteria, we have seen it's better with the galacto-oligosaccharide, and we take the lactobacillus cultures, whatever may be the species, when we give it with the fructo-oligosaccharide, indeed the colonization is better. Unless otherwise, we are looking for the colonization. So they are perfectly justified in taking a prebiotic with a probiotic. And as Dr. Seswal said, if they had not taken the prebiotic, the floor would have asked them, you have done the studies only with the probiotic, why didn't you take it with the prebiotic? That question would have come from the floor itself. And secondly, there was also a question as to, I'm not sure, I'm not able to recall, probably from you. Have you taken into the consideration the prebiotic components of the food that is generally taken? But the quantity is very negligible. The major food items that contain the oligosaccharides are either the wheat or the banana, and to some extent honey. But the quantity that is, I'm talking with respect to the fructo-oligosaccharides in particular. Okay? And the milk and other things, the content of the oligosaccharide is very less, and so the quantity that we take, it becomes very, very difficult to take them into consideration and to correlate. That need to be neglected when we are giving...
I'm sorry, I have to interrupt.
Oh, I'm sorry.
I think the one-to-one conversations you can continue at dinner or something like that.
Okay.
I'm just going to make 2 points at the end. One, an observation from the results of Dr. Satpathy's slides. There was no difference in death, but a significant, very large difference in sepsis rates. And quick calculations from your slide, it made it look like the case fatality rate, if you have sepsis, is 4 times higher in the probiotic group. And that's looking dangerous. I'm just playing the devil's advocate here. So it looks like approximately 4% case fatality rate from sepsis in the probiotic group and a 1.6 to 1.5% case fatality rate in the control group, because you had 2 times the number of sepsis cases in the control group with no difference in deaths.
But the numbers were too low.
Yes.
With the sample size, the numbers are too low.
Well, I would say this, unless you actually analyze the case fatality rates in the 2 groups and see for statistical significance, if it is statistically
My
significant...
sense is that they didn't have power for that. I mean, it was. I did see that. I mean, you did have difference of 3 deaths between the 2 groups, 3 to 4 deaths between the groups. But at that sample size, with that case fatality, you only can say that you need a larger study.
No, but I mean, the reason why I say this is one concern with the use of probiotics in neonatal intensive care units in the past has been, that was my second point, has been whether it retains the potential of colonization at catheterization sites, at intubation sites and invasive sites, and can probiotics cause invasive sepsis in extremely small babies or in facilities, because these are live organisms, or in facilities that cater to extremely small babies who have interventions being done. So there are still some concerns about using them in newborns, though the newborns provide us that point that Dr. Sazwal made, which is right. No, the point that Dr. Sazwal made is this, that if you, the reason neonates are attractive is because it seems like an age where colonization is still actively taking place. And there may be space in the niches of the gut for a probiotic or either the ones that we already have or newer probiotics to actually lead to more long-term colonization. The current definition of probiotics only includes a transient colonization. It doesn't mean that for a probiotic to be called a probiotic, you need to have lifelong colonization. You need to just demonstrate a very transient colonization. For that matter, most probiotics that at least I know of right now don't have long-term colonization. All their effects are independent of long-term colonization. But the neonates are actually a situation where we may have the potential to cause long-term colonization.
This area, whole area of microbiota and analysis of microbiota is something that is developing now. And it's only now that we have methods available to look at it and changes in it and study things. 5 years ago or 4 years ago, nobody even looked at microbiota as a microbiota. You could look at one bug, but not as a, so the assumption is that there is a lot of interest in that area, and hopefully we will see data being generated in next 5-6 years, which will basically have a holistic kind of an attitude of saying what does it do to the whole milieu as such, rather than simply that particular bug colonizing or not colonizing.
I think there has been enough discussion. We should call it close to this session. I think there are enough things to discuss during dinner now. Thank you.
Thank you, Chairpersons and Professor Sazawal and Dr. Radhanath Satpathy. Before we call this session to a close, we would like to invite you all for dinner at the Lutyens Lounge Terrace Garden. This is at 7:30 p.m. So we look forward to seeing you there in the evening. And we have the Young Investigator Poster Evaluation session from 9 to 10. So I request the jury, Professor M.K. Ganguly, Professor Satoru Nagata, Professor Colin Hill and Professor M.K. Mehra to please be there, and of course the young investigators besides their posters. And we also have a feedback form. We'd be very grateful if you could fill it and give it at the registration desk. That will help us improve our future symposia. Thank you so much for making the time for today's symposium. We look forward to seeing you at 10 o'clock here tomorrow. Thank you. Oh, sorry. Paucity of time. May I request the chairpersons to give a small memento to our speakers, Professor Sunil Sazawal and Dr. Molly. May I request Professor Batish? May I request Professor Batish to kindly give a token of our appreciation to Professor Anand Krishnan. And may I request Professor Arif Ali to please give a token of appreciation to Dr. Uma Chandramouli. Professor Arif Ali was my PhD guide, and he is the head of Department of Biotechnology, Jamia Millia. Thank you so much, and we look forward to seeing you at 7:30 in the Lutyens Terrace Garden.
इसको निकाल दीजिए, डिलीट कर दीजिए तो दोनों हमारा जो फाइल